Have you ever wondered what causes seizures in epilepsy? It turns out some of the answers are hidden deep in our genes. Let's take a quick look at how mutations in GRIN genes can rewire the brain and lead to epilepsy. I'm Gabby Cazal, a UNF neuroscience major. Epilepsy often affects key regions like the thalamus, hippocampus, and cerebral cortex. The thalamus acts as the brain's relay center, transmitting signals between different brain regions. The hippocampus is critical for memory formation and spatial navigation, while the cerebral cortex is involved in higher functions like decision-making, sensory processing, and motor control. These regions are key hubs for electrical signaling, and when that signaling becomes unbalanced, seizures form. And that is exactly the definition of a seizure. This is where it gets cellular, through neuronal communication. At the synapse, the presynaptic cell releases glutamate, a chemical signal. The postsynaptic cell receives it through the NMDA and AMPA receptors, both of which are essential for fast communication between neurons. But what happens when there's a GRIN gene mutation? Well, the NMDA receptors don't work correctly, leading to overexcitation. The NMDA receptor is crucial for synaptic plasticity, learning, and memory, making its malfunction especially disruptive. GRIN2A and GRIN2B genes are genes that code for NMDA receptor subunits. Mutations in these genes disrupt glutamate signaling, and that's a direct molecular trigger for seizures. The NMDA receptor is responsible for allowing calcium ions into the neuron, which is key for synaptic strength and plasticity. Without proper function, neurons become hyperexcitable, setting the stage for epileptic activity. Now we'll zoom out to brain circuits and understanding the imbalance in neuronal signaling. Neuronal hyperexcitability starts when there's too much glutamate, an excitatory neuron, and not enough gamma-aminobutyric acid, an inhibitory neuron. This imbalance between excitation and inhibition leads to abnormal firing and seizures. The cerebral cortex, in particular, plays a large role here as it's responsible for integrating sensory information and controlling voluntary movement. If its circuits are too excitable, it can trigger widespread seizure activity. But we are super thankful that treatments exist. Anti-epileptic drugs reduce excitability by stabilizing neuronal membranes and enhancing inhibitory signaling, while NMDA receptor modulators can correct the signaling at the synapse. Even a ketogenic diet may actually help to stabilize neuronal networks and promote metabolic changes that prevent seizures from occurring. From genes to cell to brain circuits, epilepsy is a disorder of electrical imbalance. The GRIN mutations are just one piece of the puzzle, but they show how powerful a single gene can be in influencing the brain's complex circuitry. Understanding this molecular foundation is crucial for developing more targeted treatments in the future. Thank you very much.